Stop, right?
I guess we're started. Well, hi. We are back doing a little bit of neurochemistry today. This is going to be part of a two-part lecture series. Today is going to be on transcription, and then we will follow a couple of weeks on translation. And of course, our focus, as always, is on the nervous system. Good. Here's our outline for today. We're going to focus on the issue of transcription as a form of intercellular communication. We'll set it in context in that way, and then get on. And there's rather than deal with everything in the abstract, we're also going to consider several examples today, which I think will help a great deal. So you're used to thinking about ion channels, ligand-gated, voltage-gated ion channels as the standard way of dealing with the nervous system, and it is, but then hopefully in the series on G proteins, you began to realize that there's a second type of communication, intercellular communication, having to do with the metabotropic receptors, and now today we want to talk about genetics. And we hinted at that several times. We talked about tyrosine kinases, that sort of thing. And this is going to be our focus, is on genetic modification. The time frames change. All of the ion channels are very fast. And, the metabotropic can be fast, but also can be intermediate, lasting seconds or minutes, even hours sometimes. And then you can have genetic modifications, which can be, again, hours to lifelong. So the idea here is that when these neurotransmitters are released into the synaptic cleft, neuromodulators, that they can initiate a cascade of events that lead to the regulation of production of messenger RNA and proteins. This is basically what we're going to be talking about in these 2 hours, the regulation of the production of messenger RNA and proteins in the postsynaptic cell. I got halfway through this lecture and realized I hadn't stopped to define the word gene, and I thought, well, we better go back and know what we're talking about here. So we're going to define gene as a portion of the DNA molecule, which serves as a basic unit of heredity, okay? And it has multiple components, is why it's not simple to define. You have the exon, which is the portion of the gene that's going to produce messenger RNA that's going to result in protein coding. That's the exon, right? You have the intron, which was pieces of DNA that were in between that are non-coded. They're, of course, not just sitting there as spacers. They're there for a reason, but they are not going, their base pairs are not going to be translated into an amino acid. They're going to be, these introns are largely the regulatory portions. There's another portion of the DNA molecule, which is called the promoter region, and that is where you initiate transcription. Then there's the operator. The operator is the gene segment which basically allows a repressor to bind and prevent the promoter from being bound to, and therefore prevent the messenger RNA from being transcribed off the DNA. And then you have these enhancer silencer regions, whereas the operator is always 5' upstream from the promoter region. The enhancers and silencers can be at the 5' end, they can be at the 3' end, or they can be anywhere in any of the introns as well, all right? We're going to look at all these. And then you also have to have this section of the gene called the RBS, where the RBS is the ribosome binding site. Now, the ribosomes have nothing to do today. We're not going to be talking about ribosomes today, because ribosomes are involved in translation of the messenger RNA into protein. Yet there is the site that will have to be on the messenger RNA in order for the ribosome to bind to it and initiate the protein translation. And of course, that has to be transcribed off the DNA. And so there is this RBS unit. I think it helps a great deal to think in terms of these pairs of lectures made me think a great deal about the origin of life. And I recommend strongly that you spend a little time to think about it as you go through this talk, because that's where it starts to make sense. I mean, where do you want to define the origin of life? Where is it? Is it a pool with clay that is running enzymatic activities and the sunlight and a little bit of water, and you think about these soups? Or is it are there, once you have proteins, and you can make peptides lightning into the original atmosphere of Earth, a lightning strike spontaneously generates polypeptides, not just amino acids, in fact, very few amino acids, but it generates connected amino acids and polypeptides, okay? You don't find any equivalent to that, Isama. So you don't find any equivalent of that in RNA. RNA is a very complicated molecule to synthesize in comparison to a polypeptide. And so it's likely that proteins were present before the RNA. And at any rate, what's the point of RNA? The point of RNA is to synthesize proteins. So RNA, with its genetic code, doesn't really make sense without the amino acids that it's synthesizing. So DNA is just a very fancy library. DNA is an extremely sophisticated library, but almost certainly came long after RNA. And so RNA was the first inheritable or genetic model, and a necessary molecule, you would think, in life, because if the proteins can't reproduce themselves, then you don't really have life. If they can't keep making that same protein over, then you have no heredity. All right. And if you want to, then you begin to get into a philosophical question of where is life? Does life require the ability to reproduce itself? If you say it does, then it resides in that first interaction between protein and RNA. Okay. And so we're going to see a lot of that. Okay. To the next lecture, more than this one, because that one is RNA to protein translation. But you're going to see a lot of it in this. All right. So let's return here. Here in blue, you have a portion of DNA, a gene, if you will. Here's the RNA that is initially transcribed from it. And then finally, through a series of processes called splicing, which we'll talk about next lecture, you end up with the messenger RNA, which is then going to be translated into the amino acid chain, the polypeptide chain. All right? So this is the basic idea. Notice that these introns have nucleotide pairs, so it could be GT. GT generally starts the intron region, AG ends an intron region. So again, we'll talk about this when we get to translation, but when the ribosome hits this, it's going to skip over until it hits the AG and then continue translating the RNA into the protein. I'm not going to be talking about this whole process is ancient. Okay, there's as far as we know, there is certainly only one existing process by which life forms are inherited and transmitted. There may have been other ones at some point in the evolution of life, but those did not survive. So you're going to see huge conservation in bacteria, in archaea, and in the eukaryotes. In general, I'm not going to be talking about the differences between eukaryotes and prokaryotes, okay? I think there's enough to learn without having to get into the differences, where I only focus on eukaryotic mechanisms. This is the only slide in which I actually have a bacterial mechanism. And this is where in bacteria, you can synthesize, a single gene may synthesize more than one polypeptide chain, all right? That doesn't really, it's a whole protein actually. That it doesn't happen. But I like this slide because here it shows you DNA and RNA, so transcription and translation are separated. Here's make-believe protein. And then you can see these basic pieces of the enhancer silencer, which can occur at various points. Here's the exon, right? Here's the part that will become the ribosome binding that will be transcribed in order that the ribosome will know where to bind for translation. Here's the promoter region, immediately 5' upstream of that, and an operator immediately 5' upstream of a promoter. So if you just take from here over, this is your basic gene, all right, like that. Now, like I said, transcription is the process by which you make RNA from DNA, all right? All of this is very modular, as you just saw, all right, and we went over that. As I said, this process is ancient. There's only one on the Earth right now that's involved in life, as we know, as far as we know. And there's huge conservation of these proteins. There are more than 100 proteins which are involved in this transcription translation process, which are nearly identical, but nearly identical in bacteria and in humans. Okay, you don't mess. This system doesn't get messed with very much. Okay. The ribosomal system is different, but only in detail, really. Now, in order to get from DNA to RNA, the enzyme is RNA polymerase II. All right? It is synthesized, so it is a large protein complex. So this is what I said, protein RNA interaction. All right? Now, the synthesis of the protein, so off the RNA, that is the translation process that we'll talk about next time we're together, is done by the ribosome. And the ribosome is an RNA molecule, ribosomal RNA. So the ribosome, accompanied by a lot of proteins, but the central enzymatic piece is the ribosome, which translates RNA into protein. But the transcription of DNA into RNA is done by protein. It's not, in other words, there is no ribosomal RNA equivalent for the step of transcription. Transcription is done by proteins. And so I keep pointing out this relationship between RNA and protein, okay? They are bound together, all right? This RNA polymerase II then binds to a DNA sequence, which is known as the Tata box, okay? It's a linear segment, TATA. Those are the nucleotides, and that signals the beginning of transcription. The RNA polymerase II binds to the Tata box, however, only after there are several other proteins that are surrounding the Tata box, including the transcription factors. Now, the focus of this lecture is on transcription factors. So everything else is just so you know what we're talking about. But what we're interested in is transcription factors, okay? The transcription factors are what permit selective gene transcription in specific cell types at specific times, okay? So you have this gene. This gene is always potentially ready to be translated into messenger RNA. The messenger RNA is potentially ready to be translated into protein. However, you only synthesize the messenger RNAs when you need them. And sometimes you don't want them there. So you have to be able to repress them. All right. You have to be able to, having put the repressor mechanism in place, you have to undo the repressor mechanism. You have to create a situation such that just the right moment, in just the right, think about development, at just the right moment, the cell stops dividing and starts to spin out an axon. All of that has to be done by proteins and all other sorts of things. So then you have to synthesize the messenger RNA to make the proteins at that moment. It does you no good or may even interfere with the process if you do it too soon. So you have to be able to regulate the synthesis of messenger RNA. And that is accomplished by repressors and transcription factors, okay? All right, RNA polymerase II. So this protein interacts, these proteins that are drawn to the Tata box interact and they form a complex. I'll show you a cartoon that will help in a minute. And the protein complex that's made at the site of the Tata box binds both RNA and the Tata box, which is part of the DNA, to form the transcriptional apparatus. So you have you have RNA polymerase II, you have the DNA, and you have proteins, which are linking them and making them specific. And basically, in any single organism, all messenger RNA synthesis in all cells at all times is done by the same mechanism. It's the transcription factors which say go or don't go. But other than bringing in specific transcription factors, the process is going to be the same for all cells in the organism. So that's the idea here, is that you turn the system on and off. All right. And this regulation of transcription is the basis of biologic diversity. This is what determines Rebecca from me. Okay. This is what determines humans from the strawberries that we were just eating. Okay. Is the ability to turn these systems on and off. And by the way, the strawberries and we pretty much share the same transcription and translation mechanism. There's not a lot different in the synthesis of messenger RNA between us and the strawberry here. Okay. So I love this cartoon, and there are several good cartoons that really help here. Here's your DNA. Here's your Tata box. Okay. This is the start point. You don't start at the Tata box. The Tata box is where the this transcription apparatus assembles. Okay. Here's your start point. Okay. All right. So the transcription factors bind to the Tata box. All right. They recruit other proteins. Once they make that complex, then the big RNA polymerase comes in and generates that whole transcription unit. And here you are taking right at the transcription, right at the start point, taking apart the DNA, making single strands, and can start to transcribe the DNA into messenger RNA. Okay? So that cartoon is a very helpful cartoon. There's a huge amplification that's involved here. So a single transcription factor, and there may be many, all right, we're going to look at the glucocorticoid receptors in particular. But you have, so once you initiate this, so the single trans gene, which is activated with, say, one molecule, one receptor molecule, that single gene can produce 1,000 copies of messenger RNA. And each copy of messenger RNA can produce hundreds of proteins. So then we're looking, once a gene is activated, there's a 10 to the 5th. So 5 orders of magnitude amplification of the activation of a single gene site. Okay? So this is a massive amplification. The cell is going, when you activate this gene, the cell's going to know it. Its purpose is going to be shifting dramatically. There's going to be 100,000 copies of the enzyme floating around because a single gene was activated. Activate more, you know, so, okay?
Sorry, the enzymes to transcribe, right?
The transcription step will be, so there's 1,000-fold amplification of DNA to messenger RNA, and then another 100-fold. So 3 orders of magnitude from the gene to RNA, and 2 orders of magnitude from RNA to protein. So from the gene to protein is a 5 orders of magnitude amplification.
Yeah, more enzymes with the amplification of the transcription enzymes, or the amplification of what exactly.
So you can, once the gene becomes active, so then that gene is copied many times. So you're only going to have a single gene, right? I mean, you have each cell has one chromatin set.
So that gene will be transcribed 1,000 times?
Right, that gene will be transcribed 1,000 times.
I thought there's like a picture where usually there's multiple polymerase sitting on the same gene, like a twin. Then there's multiple mRNA coming from the gene.
Yeah, and you do that, there will be multiple polymerases, and the same thing will be true. That I have some pictures from the translation lecture where you'll see the ribosomes. You can have many ribosomes simultaneously translating a single messenger RNA molecule. They're separated, if I remember, by about 100 base pairs. So every 100 to 200 base pairs, you have another ribosome. That's next lecture. And here also, you can have multiple polymerases. The basic regulation, so before we get to fine tuning, let's talk about sort of gross on or off. Gross on or off is determined by whether all these proteins can have any access at all to the DNA. Okay, the DNA is protected from all of the transcription factors, or nearly all of the transcription factors. So by the histones. All right, so the base, so let's look at histones here. Eukaryotic DNA, is not an extended chain in the cell. It is wrapped around histones, okay? So the double-stranded molecule wraps around histones, and together the DNA wrapped around the histones is called a nucleosome. These nucleosomes then pack together very tightly to make chromatin and the gene, the chromosomes. The nucleosomes, all right, block the access of RNA polymerase II to the gene promoter. And so if the DNA is in its tightly bound form around the histone to create the tightly bound nucleosome, okay, it can't be copied. So one of the first things you have to do is permit access to the DNA in its association with histones. And this is the structure. The histones are, there's another slide that will come next. But the histones are they are two, so it's an octamer. There are 4 different histone molecules, peptide chains, and they are and peptide chains. These are primarily proteins, all right? And then there's another histone which permits access, and there's this little area known as a tail. So there's a linker between them, linker region, and I'll show you tails in a minute as well. And this is nice. And here's an electron micrograph of the DNA bound to the histones to make these little nucleosomes. And these isn't an extended, they're not usually extended like this. They're usually folded into each other to make the chromosome. All right. Like I said, there are 4 different core histone proteins. You get 2 of each, so you have the octamer. The 5th H1 is the linker histone, binds them at wrapping points, and then also facilitates folding. There's a lot of folding going on. I'm going to show you another great cartoon that will come later, really help. And then there's a part of the histone which is always accessible, known as the histone tail. And that histone tail is what's going to make the histone accessible, make the DNA around that histone accessible for transcription or not. And the histone tail, you're used to phosphorylation, right? When we've talked about proteins up the wazoo, right? And protein phosphorylation has been the main mechanism that we have looked at for modification. There are others, okay? The histones also go through a series of modifications that permit access or not, including phosphorylation. Acetylation is very important. Methylation, we'll look at in a second, and ubiquitination. Right? Nice cartoon, all right? So here you have the DNA wrapping around the histone complex. Here's the linker segment. Here are the tails coming off. So that's what this slide's about, is the tails. Here's an acetylation. Here's a, glucose is put on, ubiquitin is put on, methyl groups are put on, biotin, citrulline, ADP ribose, and here's your phosphate group. So lots of different ways to modify the histones. Now, when you do, those modifications either make it easy or difficult for the RNA polymerase to access the DNA, all right? In regions that are going to be actively transcribed, you phosphorylate them. If you don't want them to be phosphorylated, look at this. If you methylate methylate either the promoter or that tail region, okay? Once it's methylated, basically that segment is long-term inactivated. So you use a gene during development and you're not going to need it in the adult stage. It gets methylated, okay? And that puts it to sleep long-term, all right? Histone acetylation, however, and phosphorylation are quicker. They're much more on or off. And we're going to be looking at this process through the transcription factors, because a lot of the transcription factors, including the ones we're going to talk about, act through the process of histone acetylation as well as phosphorylation. Right? And here you can see, I'll just give you a time frame of this. We're going to see others, but these growth factors, neurotropic factors increase that are involved in memory. We're going to see a lot of messenger RNA transcription in memory. That when this occurs, they return to baseline within 24 hours. So you get roughly 24 hours to solidify your memories through these mechanisms. Yep.
How much do we know about the ability of patients to
So that's a point that I was going to come back to later. No, it's perfectly fine, because here's the thing. You're going to be looking at proteins. The proteins are the transcription factors are proteins, which means that they had to be translated off a piece of transcribed RNA. Okay, so there's this circularity. So the proteins are controlling the DNA, but the proteins are synthesized themselves, and they are subject to the same regulatory process that they are engaging in. So there's this huge circularity in this system where the DNA is being transcribed to RNA, the RNA is making proteins, the proteins are functioning to regulate the DNA. And in some cases, we're going to look at SOX too. I know Dr. Bradley said he talked to you about the HOX gene, right? So there's no other purpose to these proteins except to serve as transcription factors. They don't do anything else, but they have to be synthesized. So something created them. Some signal to the cell signaled the need for a regulatory protein to be synthesized. That then the regulatory protein goes back and regulates the further transcription of other downstream proteins. So there's this highly regulated, incredibly complex system. All right, let's just keep letting it play out. All right. So now some technical information about transcription factors. Okay, now we're focused on transcription factors, and will be for the rest of the talk. All right, you'll hear the term cis regulatory element. So cis means it's on the same side as the transcribed DNA, right? So in other words, cis regulatory elements are on the same strand of DNA as the sense section of the exon. All right? So in other words, if here's the double strand, this is the one that's going to be synthesized, all right? Anything that's on this strand is known as cis. Okay, it's on the same side as the DNA segment that's going to be transcribed. That's cis. All right? They are usually at the 5' flank. So a cis regulatory element here, this is what they're known as, regulatory regions or regulatory elements. You have the promoter. All right? The promoter is a cis acting. Okay? So was the operators are cis acting. All right. These are in other words, these are on the same strand as the sense, as the part that's going to be translated. So we saw that the promoter is always immediately upstream of the transcribed portion of the gene. The operator is always immediately upstream from the promoter. But another group called enhancers, which can work in either direction. They can be enhancers or silencers. Those can occur upstream, in the middle in an intron, or downstream. So enhancers, okay, get to occur at numerous places, but the enhancers also are cis-regulatory, meaning that they're on the sense strand of DNA. But they are going to bind other factors. Okay, so then cis is on the same side, trans is either on the opposite side or far away. Okay, transatlantic. Right? So trans elements are not on the sense strand of DNA. Trans may not even be on the same gene. They may not even be on the same chromosome. They can come from totally someplace else, but they act by binding to a cis segment. So the transcription factors, okay, are all going to be transacting. They come from far away, and they all are going to interact with the cis regulatory element in order to provide access of the RNA polymerase II complex to the gene that needs to be transcribed. So here you go. Here's the DNA, right? Here's the DNA. Here, the components of the RNA complex are just floating around. Some of them are bound, some of them are not bound. This thing is in no way ready to transcribe DNA. Now, they come together, usually under the influence of some other proteins as well, all right? And they have assembled, but they cannot transcribe. But now along comes a series, and we're going to look at this. This is important. not just one necessarily transcription factor, maybe multiple transcription factors are needed and come together, bind to the RNA polymerase II, and together now they can transcribe the DNA to make RNA. Okay? Now, these transcription factors can, like I said, can be activators or repressors, all right? They are transacting because they're not part of the regulated gene, okay? There are lots of groups, but they all have, they're all capable of combination of protein interactions and have DNA binding domains. They have to be able to do both. Now, this is really critical here to understanding transcription factors. All of the transcription factors are dimers, okay? They have sections of the transcription factor. So each peptide chain has an area that allows it to dimerize with another transcription factor. Now, we're going to look at them. So you've heard of zinc fingers and leucine zippers. This is what we're talking about. These are the dimerization sites, are the zinc fingers and the leucine zippers, okay? But here's what's really amazing about this system. It doesn't have to be a homodimer. If I'm a glucocorticoid receptor, I do not have to dimerize with another glucocorticoid receptor. In fact, I'm probably not going to dimerize with another glucocorticoid receptor because I already got one of those. What I want is a different transcription factor that will make a ubiquitous signal like a steroid totally specific for a cell at a particular moment in time, okay? And the same glucocorticoid receptor at one moment in time may turn the gene on, and the other moment in time may turn that same gene off, okay? Depending on what other transcription factor it is bound to. So that is basically the essence of this entire talk, okay? So here's the idea, is that you can make these heterodimers, and the heterodimers allow incredible specificity through time and space. Here's your leucine zipper, here's your zinc finger. There are others. This is helix turn helix, there's a helix loop helix. These are basically dimerization structures. All right. Now, here's the other thing, is that these same cis-acting elements can occur in multiple genes, so that the transcription factor, which binds to the cis-acting element, may turn on multiple genes, or quite frankly, may turn on one gene, and with the same cis element, may turn off another gene, okay? So this is where your specificity is starting to come from. You have the gene that's going to be transcribed. You have the cis-acting elements that are allowing them to be regulated. And now you have these transcription factors, which of course are proteins that have to be synthesized. And if they were proteins that have to be synthesized, then this whole process of messenger RNA and DNA and activation of the DNA in order to make the transcription factor happen. All right? The specificity arises from where the cis-acting element is in relationship to the promoter and the identity of the protein partners that make up the heterodimer, the protein partners. I love this picture, okay? So to really focus on this picture, because I think up until now, and I think most of us have the notion of DNA as a linear molecule, right? But it isn't, okay? When it's working, it folds on itself. So you say, well, how could the enhancer all the way up here, nowhere near the promoter region, have anything to do with whether the gene is promoted or not? Is it because it prevents the polymerase from going on, or what's going on? Well, look at this. So here you take these activators to the enhancer region, all right? And at the same time, you bring in yes, it's called the DNA bending protein, not binding protein, bending protein. Now you bend the DNA, maybe this is why we need introns, you bend the DNA so that now this enhancer region is actually immediately next to the Tata box and can either favor or inhibit binding of the RNA polymerase complex in order to, there it is, And here's the final product. So there you go. Okay? So you have to get rid of this notion of an extended DNA molecule and realize that such an extended molecule is capable of a lot of motion. Okay? And you can regulate that motion. Okay? And bring elements that are far away immediately adjacent to it, to the promoter region. Okay? So I think this is an absolutely great cartoon. And I think that Histone cartoon and the other one before it, I think just really give you a huge amount of information. So that's the basic concept. And let's look at 3 examples. We're going to look at 3 examples. I want to look at the glucocorticoids, or are we short of time? Glucocorticoid transcription. Right? I assume you're going to find this fascinating. We work with steroids all the time. So there are 2 high-affinity intracellular corticosteroid receptors, type 1 and type 2. Type 1 is a mineralocorticoid that binds either cortisol or aldosterone. Type 2 binds dexamethasone. So have any of you ever tried to treat gout with dexamethasone? So I did. I had a person who was salt overloaded, and so I didn't want to use Solumedrol, and so I gave him dexamethasone. And after 2 days, he didn't get better. He had, which I've never seen really with Solumedrol. And I asked rheumatology, I said, why is the dexamethasone not working? And they just shrugged their shoulders and said, I don't know, but it doesn't. Okay, well, now I know why it doesn't. Okay, because the dexamethasone receptor, okay, is not the one that's controlling the inflammatory process related to gout. 2 different receptors. Okay, so is there a difference between Solumedrol and dexamethasone now when we're doing things with MS or tumors? Or the answer is there are. There are 2 different receptors altogether. Okay, I'm going to focus on hippocampus, not a lot, but I just want to give you a sense of the importance of learning. And so the glucocorticoids are present, the steroids themselves are present in high concentrations in hippocampus. Both of the, now we're looking at the glucocorticoid, at the corticosteroid receptors, not just in and of themselves, but they are an example of a superfamily of ligand-activated transcription factors, which are hundreds of receptors. Okay, so this mechanism that we're going to look at with the corticosteroids is heavily utilized by the cell for things as diverse as estrogen, retinol, the thyroid hormone, of course, steroids, that sort of thing. They have these receptors. Now we use the term receptor, but you need to get out of your mind the membrane receptors. These are not membrane. These are cytoplasmic receptor proteins. They are receptors because the hormone is going to be intracellular, okay? They're not on the membrane surface, these receptors, okay? These are intracellular receptors floating around relatively speaking in cytoplasm. So each receptor has 4 distinct regions, okay? There is the N-terminal region, which is the one that's going to interact with the DNA in order to activate transcription. There's the C-terminal, which is the hormone binding region. And in between is the DNA binding domain for attachment to the DNA and a nuclear localization signal. We're not going to talk about that today. But in the lecture on translation, we will talk about the importance of this nuclear localization signal because, look, right, you got the signal is hitting, might be start at the membrane. We're going to talk about cyclic AMP. Might start at the membrane, might not. Definitely is going to be in the cytoplasm with its receptor. That receptor is going to control the DNA transcription through an interaction with DNA. Therefore, it's going to have to get into the nucleus, okay? The nucleus is going to transcribe the DNA into RNA, but the RNA translation step is going to occur out in the cytoplasm. So this complex has to get back out. So we're shuttling in and out of the nucleus, which is why I wanted this lecture before the one on frontotemporal dementia, because the frontotemporal dementia was all TDP43, okay, which is a protein whose purpose is to move these things as a chaperone in and out of the nucleus, okay? So that's where we're going with all this, but they all have this 4 domains, the active domain, the hormone binding, the nuclear localization signal in and out of the nucleus, and DNA binding. And this is a nice cartoon that shows you the N-terminal domain and the DNA binding domain and the ligand binding domain, and then the nuclear localization signal. All right. So in the nucleus, in the cytoplasm, the receptor. Now let's just take the corticosteroid receptor as our example, and we understand that it's an example of many other types. So these receptors are associated with a multi-protein complex, okay, that contains heat shock proteins. So you probably heard about heat shock proteins. They are stress response proteins. The corticoid receptor is bound to heat shock proteins in its native quiet state. And the receptor is basically not doing anything when it's bound to the heat shock proteins. However, when the corticosteroids come in and bind, okay, to the receptor, all right, so the cortisol, dexamethasone, all right, then the receptor is released from the heat shock protein and is now capable of translocation into the nucleus, right? If it can't get into the nucleus, it can't do its job, which is to activate the DNA. So while it's bound to the heat shock protein, it's inactive. Binding of the steroid then releases it from the heat shock protein. And then in the nucleus, the ligand-bound receptor, which is a transcription factor, a trans-acting transcription factor, then binds to a cis-acting element on the strand of DNA that is going to be transcribed. Okay, and that's the beginning of it. For the glucocorticoid receptor, there's a particular base sequence, which is how it knows where to bind. This sequence, however, can be anywhere in the gene. This is an enhancer type, and so it can be 5' end, 3' end, or in an intron. Okay. Good cartoon. Here's the steroid. Here it is diffusing across the cell membrane. Here it is binding to the receptor complex. Here's a heat shock protein. The binding releases the heat shock protein. The receptor is now activated and loose. It is transported into the nucleus where it can bind to the enhancer, the cis-acting regulatory element of the DNA, leading to RNA polymerase II formation and transcription of the DNA. Okay. Now the thing is, as we said, these corticosteroid receptors don't make homoreceptors; they make heteroreceptors, so they can bind to other things. So now the steroid might be, even if the corticoid receptor is active, it may not lead to transcription, okay, or may lead to some transcriptions, but not others. Only when it's bound to a second transcription factor does it now become active. And so you can make the corticoid. Here's an example where you have this. These are proteins that are being synthesized, and they require heterodimers of the steroids. So the steroids alone don't do them. And they act, these transcription factors act through histone deacetylase to give the polymerase II access to the DNA. All right. Here's a picture. This is a homodimer of AP1, okay, with DNA. I just put some cartoons in at the last minute to help. So just quickly to go through the cyclic AMP. Cyclic AMP here, we don't need all these slides. Cyclic AMP, we've been through this, right? Cyclic AMP is bound to the receptor, right? It activates protein kinase A. You probably don't remember, but active protein kinase A is free of its regulatory units. It's an active enzymatic subunit. It was translated, okay, it is not translated, it is carried into the nucleus, all right, and its job, of course, is to act as a kinase, all right, so it's going to phosphorylate a series of proteins, all right. How does it get into the nucleus? And once it's in the nucleus, what does it do to allow the DNA to be transcribed? So here you go. All right, here's your receptor agonist. Here's the G protein complex, right? Here's the adenylyl cyclase. The cyclic AMP is synthesized. It activates the protein kinase A, which is released from its regulatory units. The protein kinase A makes its way into the nucleus, phosphorylates a protein which is cyclic AMP response element binding. So CREB, okay, is intimately connected to cyclic AMP. So here's the entire signal from any of these receptor agonists through cyclic AMP phosphorylation of the CREB, and now leading to transcription. Okay? These are just, this thing just plays out multiple ways. And lots of evidence for involvement in learning in simple organisms. Okay, so just a couple of pictures. Here's a heterodimer binding to DNA. This is looking down the long axis of the DNA. This is just the kind of thing where you can get heterodimers forming, and depending on some of the heterodimers may actually be an inhibitory factor, and don't let it bind. But this inhibitory factor only binds with this particular factor A and doesn't bind to B or C. So this is just an infinite number of ways of doing this. Again, it's very late. I want to just point out a couple things, going back to the interaction of protein with RNA. All right. Here, in order to recognize a 7-base DNA stretch, all right, so the transcription factor has to bind to this codon, this 7-base sequence, okay? It takes 79 amino acids in a protein to interact with these 7 base pairs, okay? So this is not an efficient system, okay? This is a somewhat complex system, which again, when you think about the evolution of life, realize that these things they're not spontaneously here. Okay, these are things that had to evolve over billions of years. Okay, and once they evolved, they never went away. Okay, they stayed. Right, only one system on Earth, basically. Okay. SOX2, again, rather than HOX, I'm using SOX2, and just very quickly. Okay, SOX2 keeps progenitor cells active as progenitor cells, okay? And if you inhibit the SOX2, then the cell exits the cell cycle. So there you have a simple mechanism for, if you think about the ventricular zone where neurons are being generated, when all you have to do is inhibit the SOX2 and bang, that's it. The cell's going to exit the progenitor cycle and set up shop as a neuron. All right. And it does it in coordination with multiple other transcription factors, so that different places in time and space. Okay, I really think we sort of have enough. The message is when these transcription factors allow, so you can have, for instance, gradients of how does an axon know when it's gotten to the muscle cell? How does it know when to stop growing? Literally stop and create a neuromuscular junction. How does it know when to do that, right? Because the protein, the muscle cells are synthesizing signals, right? And they're releasing signals into the extracellular space. And when the growing terminal, what do you call it, the axon, the growth cone, encounters that environment around the muscle fiber, okay? Then it senses those signals, which are then change the messenger RNAs that are being transcribed. And suddenly it finds itself now making neuromuscular junction proteins, okay? As well as not blowing past its target. So it has to turn off the genes which are causing it to grow, stop the growth, and extend out the neuromuscular junction. And of course, what happens ultimately in the nervous system, and we'll spend a fair amount of time on this next year, but ultimately what happens is that all of this, the segmentation of the nervous system and its specialization is occurring because of the notochord, the interaction of the ectodermal plate with the notochord. And it is that interaction then which causes cranial nerve 2 to develop, or cranial nerve 5 to develop, or the otic cup to develop, or everything else. It's all in relationship to the notochord. All right. A quick cartoon just showing you heterodimers, and the heterodimers are binding to different regulatory systems. See, part of it is the same because it's SOX, but then part of it is different because the other transcription factor is different. So only when it finds those two signals Does it know that it should set in motion transcription, which in this case is very different than this case? All right. And all of this is being now manipulated. This is sort of the cutting edge of pharmacology. I mean, that's basically the idea, is what you're doing is developing drugs to regulate the kinases, regulate the acetylation, and also to use parts of what's known as the transcriptome. And the transcriptome is the transcription factors, along with their regulatory genes and other proteins. And so this whole thing is known as a transcriptome, and you can modify that. And you can modify it in lots of different ways. You can use microRNAs, bind to RNA segments, inactivate them. You can use small molecules to favor acetylation, to interfere with it, to methylate the histone, turn things on and off. And that's basically what's going on. Okay. And that's it. Yes.
Yeah. So I was going to ask, basically, without trying to extrapolate this way too far, but I think you actually just kind of touched on it at the end there. Would you say that this is kind of like the basis of epigenetics and the field of epigenetics and potential future modulation?
So yeah, like epigenetics refers to the modification of all of these steps of transcription and translation in order to result in more and more selective protein synthesis. So a lot of it is occurring at the histone level, and all of that is known as epigenetics. But then in the next lecture on translation, you'll see that also can occur at the translation level as well. You can modify. The first thing you do when you create the messenger RNA, by the way, again, that's more or less where we'll begin, is we're going to start with a piece of messenger RNA. The first thing it's done is it gets a poly A tail, because without the poly A tail, then the ribosome doesn't bind to it in order to synthesize the protein. So that's going to be the next step, and this will make a lot more sense once we have translation in view.
So, like, sort of like the example you brought up about the neuromuscular contraction being created. What I always never really understood was like that those molecules being produced by like the muscle in that instance are sort of like diffusing across, you know, sort of
right
at a high point of concentration near where they're made, and then less and less concentrated as they diffuse away.
Yeah, as well as on their surface. Right,
but yet like any organism has very often like very demarcated on and offs of where things are made, and when my eye develops and then stops and then starts something else. And yet on that molecular level, those morphogens are in a gradient. So what mechanism is causing a delineation when the basic thing informing the process is a gradient? So
I would say it's the same, to me, it's the same principle as localization in the nervous system, what I call axial localization. And that is, you have to have a second element, okay? You have to have at least two. Because like you said, if you only have one and you're working along a gradient, then, okay, you might go down the gradient, you might be repelled by the gradient, but that's not a very discrete, okay? So couple it with a second message, where the second message is being built more discreetly, and it doesn't have to broadcast very far, maybe a membrane receptor, so that you might attract the growth cone toward the target because it's going up the concentration gradient, right? But that's not enough. But it doesn't do anything. It doesn't form that synapse, that